Kennedy, Jalen, Claire,
Hello.
how's everything going?
It's going all right. You?
You know, I'm doing okay.
Good.
My girlfriend's mother made us some handmade masks for the coronavirus.
Oh, nice.
Life could not be better. I have a mask. It's got a little pouch to put a filter in there. How are you guys holding up?
Doing well.
You should have. Well, let's see. Who are we still missing? Oh, we're missing quite a few people. Luke, where in Texas do you live?
Little town called Floresville, just south of San Antonio, about 35 minutes south.
South of San Antonio. Okay.
It's about an hour and a half away from Austin.
In the area where I need to go gecko collecting and can't.
We're in Galveston.
No, not Galveston. Well, Galveston also. I guess they're about to close off Galveston. They're about to put roadblocks on the causeway going over to Galveston Island. But also I need, Gonzales is one of the places where I need to collect. Port Aransas, Brownsville is further south than you. But that neck of the woods.
Yeah, Gonzales is about 2 hours. We sell cows there.
East of you?
Yeah. East.
Yeah. All right, everybody is here. Is that correct? So you guys all should have received your Brody literature summaries in the email from me this morning. It says from Reprographics printer. It looks like spam, but it's actually scanned from the Xerox machine here in Marston. So open that up and look at what you did. And then if you need to make corrections, send them back to me by Wednesday. I am currently working on your exams. I'm about halfway through them. So I'll probably have them finished up later on today, and I'll scan them and send them to you when I have them done, which will probably be this evening or tomorrow morning, depending on how quickly I get through them and whether I finish them at home or if I finish them here at the office. So that will be that. We're talking about the McClintock and Ewitz paper today, so somebody needs to share a screen so we can all see what's going on and take over the reins.
Cool.
In your participants tab, if you go down to participants, if you have that open on your screen, you can either leave your mic muted and unmute yourself with the space bar, or by clicking on the mute on the microphone icon in order to just interrupt. Or if you want to be in the participants tab, you can raise your hand and it will appear in the participants tab, and then Luke and or Haley can call on you, whichever one works for you guys. So take it away.
Can everybody see my screen?
Yep.
Yes, okay, cool. So Luke and I are going to be talking about McClintock and Ewitz, 1996.
Hold on. Oh yeah, okay, never mind. We are recording. Go ahead, sorry to interrupt.
Okay. Great. There we go. Can somebody kind of tell us what was previously known, where they wrote this paper, what the renovation was?
You broke up from my perspective. Can you repeat that?
Can anybody tell us what was previously known before they wrote this paper?
I believe they knew that if males did not communicate properly, they would either be, or they could be attacked or mate with the female, depending on their communication.
Okay. Yeah, anything else? There were a couple things they mentioned. That was one of them.
They talked about like the tactical or strategic design of these signals, and essentially like the strategic design is kind of more to do with the response and how attractive it is to females, and the tactical is more with like, is it like effectively produced and transmitted through the environment? And those are kind of the, you know, categories of study that have been looked at, but less the tactical category.
Okay.
So two different ways of thinking about a signal.
Can anybody tell us what Darwin's theory of evolved traits was?
If you recall, there's several components of this.
It was whether like the signal was successfully generated, if it was successful for reaching the female, whether the signal was received by the female once it reached her, and the decision of the female to act on the perceived signal.
Yep. So those were the 4 factors that Darwin's theory talked about that were impacting the female preference. And then, yeah. Luke.
Just so you guys know, we're going off of speaker notes, so we have cues on who talks when, because it's kind of hard to do it not in person. So if we have some interruptions, don't get mad at us. So Alexis kind of mentioned the tactical design. I think you did a good job of explaining that. Does anybody else want to jump in on ideas or thoughts that they gathered while reading this? I think Alexis did a pretty good job of explaining what it was.
One thing that I thought was important to it is that in situations where tactical design is important, there's a strong select on courtship behavior, because ineffective signals during courtship could be lethal to one of the parties, in this case, the male. So I thought that was important to note for the tactical design as well.
Is that more of a tactical issue or a strategic issue?
In the paper, they label it as a tactical issue.
Yeah.
Okay.
On the top of page 168, that paragraph that follows in from the previous page, it talks about strategic design, and then it goes into tactical, and it just says that in mating systems where the consequences for ineffective communication are severe, for example, cannibalism,
Gotcha.
potential mates, selection on tactical design may be strong, because in such cases, an ineffective signal may be lethal.
Okay. All right.
So microhabitats, if you recall, they were different. Does anybody remember the 2 microhabitats of the spiders? Kind of a specific question, but you can list the, yeah, there you go, Haley. So this is the Acreata microhabitat. This is an actual picture of the Cincinnati, Ohio Nature Sanctuary. It's a deciduous forest, it's dry leaf litter. This is just an image so you guys can get a visual feel for what it looks like rather than just reading it on paper.
This species is by far the most common wolf spider species that you see out at Martha Lafitte Thompson if you've spent much time out there.
And then the Acreata, this is an actual image of what the spider looks like. And right off the bat, you notice the tufts. And on the right there, you can see where they're located. as well. So just to kind of see what they look like. I don't think the literature gave any images of what tufts look like, so we thought it'd be helpful for you guys to see.
And then the Ravnrai species were also used. They also used the Cincinnati, Ohio Nature Sanctuary, but they were more common in a floodplain type forest where the rivers would kind of flood over the grounds, and there was a lot of more water that compacted the leaf litter down. So it was not as, I almost want to say not as fluffy kind of, but like the leaves weren't as dry, and it was a lot flatter ground for them. And then these are the Ravnrai species. So on the right here, this kind of grainy photo, it shows very, very small, basically no tufts or bristles on the foreleg. And this image is actually from another paper that was done later on called Stratton, 2005. And she also studied the same spiders, just a little more in depth, which we'll probably talk about later. And then does anybody remember the two hypotheses?
Like the key one was the pre-existing bias hypothesis, which basically says that if females had a preference for the tufts before the tufts actually evolved in males, then like that would represent kind of this pre-existing bias.
Yep, that was one of them. And does anybody remember the second kind of big hypothesis that they had?
I think the Acreata.
I guess like maybe like just kind of did the tufts actually work as a signal to the females?
And that, yeah, and that relates to species recognition. Yeah, so you're on the right.
In the abstract, it says something about the hypothesis that these tufts influence female choice.
Mm-hmm. So as far as the methods, can somebody give us just a brief rundown of what they used and what was kind of done?
From my perspective, they used both of these species. They took videos of the males doing their kind of courtship displays, and then took those videos and manipulated them. And then they put the females into a transparent arena with their little televisions on the ground where they showed the videos of the males that they have either done nothing to or like added tufts or taken away tufts completely or enlarged tufts by 25% for the Esocreata, and then showed those to the females and basically just looked at, their latency, their general proportion of receptivity, and then they also labeled their receptivity on like scale level.
Yeah, that was a hell of a description.
Okay, so that's part of what they did. What about the second part, somebody other than Kennedy?
They used live male spiders and they categorized them as having like large, medium, from the Ocreata species, and categorized them as having large, medium, or small tufts, and then kind of presented them to the females and recorded similar numerical response scores.
Anybody else want to add anything else?
About the phylogeny.
Somebody other than Kennedy.
They tried to construct a phylogenetic tree that determined whether or how the tufts evolved, whether the tufts evolved from a single common species or whether they evolved in multiple separate instances as an amplifier for attractive mate characteristics, if you will.
Does anybody know how many species they used when they constructed their phylogenetic tree?
6 and an outgroup.
Good.
Yep.
Did anybody catch how many species at the time there were for Schizocosa?
More than 20.
Yes. All right, Luke, you ready?
Yeah. So before you click to the next one, so this is a Watchman, and I had no idea what it looked like. I've heard of a Walkman before, but I've never heard of a Watchman.
It's such ancient technology.
It's before our time. So I looked up an image of one, and I saw Dr. Kowinski smile because he knew it was coming next when we were talking about methods. But there's a screen here, and my first thought was when I saw the screen was, well, how good is this spider image going to look? You know, is it going to look choppy? Is it going to look, not clear? And then,
Well, and I think I think if I recall correctly, because I was alive when the Watchman was very popular, and they were really expensive. So that if you were cool and happening, you like owned a Watchman, and if you were poor, like a graduate student, you owned a Walkman so you could listen to music. But I think this was a 2-inch diameter screen, so these things are actually really small.
yeah, it's smaller than our phones, to put it in perspective.
Yes, this was also pre-cell phone days.
Oh yeah, it's crazy. But we looked up a video, and we're going to show you a video next. So Haley, if you want to go ahead and click on the video.
This video is actually from the UIT's lab. It's from his actual lab as an example of what they had done for this paper.
So you can see the male doing his courtship dance, and that
Yep.
Is that a female or is that a male? Hard to tell. I guess that's a female.
So if you notice this male,
Did you just see what she did? Run that back about 10 seconds. Do you see her just flatten her body?
mm-hmm.
Was that what you were about to point out?
Actually no, but sure.
Oh, okay. I thought I was stealing your thunder there for a minute.
No, you weren't. So here, I think.
So she's up on her legs. She's looking at the male. It's not really a male. There, she just flattened her body. Yep. Now she comes back up on her feet because the male is not responding to her flattened body. There she goes, flattening her body again. And wondering, why is this male not catching my clues? And so you notice also that that male is just on a loop. They've just looped this video over and over again. Oh, now she's raising her abdomen. I should do my David Attenborough voice.
You still got a little bit of time.
10 seconds.
Oh yeah, see, there she is raising her abdomen again. So if you guys go and look at table 1, these behaviors that signal female receptivity, flattening the body down to the ground and also raising the abdomen are signals that the female sends to the male to indicate receptivity. And both of those characteristics were illustrated in that particular clip.
So this is kind of just a drawing of basically what they did. So they had spiders that had no tufts, like in this first image, and if it was Ocreata, it would wave its legs. If it was Ravneri, it wouldn't. And then they had the control Ocreata spiders that had the tufts, and they would wave their legs back and forth. And then they also had spiders where they had enhanced the tufts on the Ocreata and they kept their leg waving. And then they added tufts digitally to the Ravneri spiders. And of course, they didn't wave their legs because that is something that's common in the Ocreata spiders, but not the Ravneri spiders.
Hey, Caleb, you want to explain this scoring method to me? No.
The scoring method.
Yeah, you just kind of want to walk us through that.
So basically, the higher value is the more likely it is that the female wants to mate, and the lower value would be like a no response or predation display.
Yeah, pretty simple. This is their scoring method, and like Caleb said, the higher the better. And 3, as you see the asterisks, there's 4 types of behavioral patterns that will result in a copulation. Typically, that will occur after that.
Doesn't necessarily mean that there's going to be a copulation, but generally copulation doesn't occur unless you have one of those.
Yeah.
So in the video we watched, there was kind of the settling behavior as well as the lowering of the abdomen or cephalothorax and the raising of the abdomen.
Yep.
Can someone who hasn't spoken describe or tell me what figure 1 is talking about?
It's talking about the number or proportion of receptive females under certain stimuli or conditions. So if you look along the x-axis, it shows control, no tufts, enhanced tufts, and that's all for S. ocreata. And then control tufts added in a blank screen for S. ravneri. And then above the key or the legend, it says female S. ocreata or female S. ravneri. So they're displaying each condition for each female type. And the y-axis is showing you the proportion of the females that responded or that were receptive, like whether there was a presence of receptivity or there was no receptivity for each condition.
Yep. Can a different person tell us what the letters mean above each column?
The letters show you which groups are statistically equivalent. So like in the top, all of the black bars are equivalent to each other. And then the untufted Ocreata is then equivalent to the Ravneri control and the tuft added control.
Yeah, so they use these letters to show where the significant differences are. So if you look on the S. ocreata on the top, the control, the no tufts, and the enhanced tufts were all pretty much the same. There wasn't any significant difference between those 3. The one in the middle right here that has no tufts was also pretty much, it was similar to the control for the Ravneri. However, the control for the Ravneri was significantly different from both the male Ocreata control as well as the enhanced tufts. And then down here, it's pretty much the same thing. So if any of these columns has a B on it, then that means that they're not significantly different. Likewise for A, if they have both letters, then there's no significant difference across any of them. So the enhanced tufts on Ravneri was not, for the male Ocreata right here, was not significantly different from any of the trials.
This might be a question for Paul, but you guys might know the answer to. Would it be right to call the A-B categories kind of intermediates? Because I'm still confused about how the A-B, like the notation of the A-B versus A and B. Like I get it, but I also don't get it.
So the first 3 dark bars those individuals are not different from Ravnarai with tufts, nor are they different from a blank screen or Ravnarai without tufts. So those aren't different from anything else in the figure. Now, Ravnarai without tufts is different from Ravnarai with tufts. And Ravnarai with tufts is better than a blank screen. Does that make sense?
So is that saying that the Ravnarai, the three black bars, is that saying that none of those are significantly different from the blank screen either because they all have B's?
Yes, that is true. Yep, that's exactly what that means. So when you give a Ravnarai a Okriata signal, an Okriata stimulus, they respond to that stimulus no differently than they would a blank screen, statistically speaking. But if you give Ravnarai stimulus some tufts, they respond much better to that than they do to Ravnarai without tufts.
I guess like my still concern is just like, I mean, just the write-up where it's like you kind of get into the weeds of explaining what's significant versus not significant in comparison to each other. So it's like, would it be an effective use of language to call that an intermediate receptivity or not?
I mean, if being intermediately stimulated means that you could just as easily be watching snow on the screen. Because, and I can't point to it, but the blank screen has a really, really low proportion of females responding. Go down to Ravnarai. Really low proportion of female Ravnarai responding, but that's not statistically different from any of the other bars except for Ravnarai with tufts added. So the first column, second column, third column, fourth column, and last column are all not different from one another. So I think a more productive way of writing about this in words is to think to yourself, how does Okriata respond to Okriata signal versus Ravnarai signal? And then within Okriata signal, do they respond to different kinds of tufts? And Okriata doesn't really respond differently from, Okriata females don't really respond differently to the various Okriata signals. And if you throw tufts on Ravnarai, that's also not different from an Okriata signal for an Okriata female. But Okriata is generally different from the Ravnarai control, except for the Okriata that has tufts removed, which then probably makes it look quite a lot like a Ravnarai. So I think the way to think about it is think about which females are responding versus which stimuli they're responding to. Okriata responds equally well to all the Okriata ones, all the Okriata stimuli, regardless of whether it's controlled tufts, no tufts, or enhanced tufts. They also respond equally well to Ravnarai with tufts. The only place where there's any sort of weird difference is that they respond less well to Ravnarai control, and they respond less well to blank screen.
Okay. I guess
That makes sense?
yeah, the paper just kind of makes qualitative statements about intermediates. The paper in the actual discussion of these figures doesn't say that Okriata responds well to no tufts, like as well as to enhanced or regular tufts. It calls that kind of an intermediate response, even though it's not statistically.
It's because they want it to be true, even though they don't have the numbers for it to be true.
Okay.
And you've seen me in senior research. You know what I say to those kinds of things? If you don't have the numbers, you either need to go and get more numbers, or you need to stop interpreting things that aren't supported by the numbers that you have. Because they look intermediate, but they're not statistically different from one another. And so it is striking that over 50% 50% of the population that they tested responding to those. So in the Ravnari panel, point at the Ravnari panel. So that's around between 50 and 60% of Ravnari responding positively to Ocreata signal. The idea that that is not significantly different from a blank screen that is down somewhere below 10%, what that tells me is that you just have a really weak test. And why do you have a really weak test?
Generally because you don't have a large enough sample population.
Yeah, you don't have a large enough sample, and the test of proportion responding is actually a really crude measure. So you don't have a very precise measure of receptivity, and you don't have a very large sample size.
Can I ask one more quick clarifying question?
Yeah.
Can you use the letters just threw me for a loop when I was reading this for the first time. Can you compare graphs within the same figure? So can you say that the control Ravnari in the bottom graph is significantly different than the control Ocreata in the top graph? Can you
No,
compare?
because the analyses were done separately for Ravnari females and Ocreata females. So
Okay,
the
so you
letters
can-
only correspond within a figure.
Okay, so within one graph or the other graph, not necessarily within the.
So when you look at when you look at the figure caption, they only had 21 Ocreata and 18 Ravnari females. That's really not very much for doing these types of analyses. And in the other panel, you only had 24 Ocreata and 20 Ravnari. Once again, pretty small sample size. Because you're just placing these individuals on a 3-point scale. So what they're not showing you, which is something I wish they would have, is rather than putting the little spider icons on top of the bars, what would have been really nice to see?
Standard error bars.
Yeah, a 95% confidence interval. And so what's happening is there's probably a lot of variation from individual to individual. And so what looks like a difference when you just look at the plain bars isn't statistically detectable because there's just a lot of variation from individual to individual. And that's frustrating because they want to say more about the data than they can. And it's frustrating from a reader's perspective in that it's impossible to just look at the bars and know, well, how much variation is there around those average values? Which is why I'm a huge advocate of putting meaningful inferential error bars around means when you plot things. Because this really points out why that's important to see. Sorry, I feel like I've now hijacked your discussion. Continue.
So figure 1, I don't even remember who it was. I think it was Jack. Said that
it was
this
Jack.
is okay. This is just kind of the as it says here, the proportion of females displaying sexual receptivity. Can anybody kind of explain what figure 2 is? Because if you at least look at the y-axis, figure 1 has the proportion of receptive females, figure 2 has the mean score. So can someone, not Alexis and not Kennedy, talk about figure 2 and not Jack?
And not Jack.
And no Jack either. Yeah.
That kind of leaves Jalen, Claire, and Caleb, right?
So it's basically just like the average score between each stimulus that the female, like S. Ocreata or the Ravnari were shown.
Yes, and so those scores we showed you actually on this slide.
Yeah.
So figure 2 is just graphing the receptivity of those scores on the previous slide. Again, the letters signify the significant differences and everything. We don't have it shown on this one because I whited it out, but did anybody notice on the S. ravneri panel that the control had a letter C?
Yeah.
Yeah, noticed.
Yeah,
Which doesn't make a damn bit of sense, actually.
yeah. We pretty much decided it was probably a typo. It could have been, they could have meant an A, but more or less it was just a mistype on the graph. Does anybody have any questions about these two figures?
So the receptivity score in figure 2 is just the average of the behaviors shown that are outlined in table 1. Is that correct?
Yes.
The average of the maximum that an individual reached.
Okay.
Because flip back to table 1, Haley. Yeah. So obviously, you have to go through 1 on your way to 2, and you probably exhibited 2 before you got to 3. So it's basically what's the maximum level that you reached. Now, in theory, you could jump from no response to lowering your cephalothorax and raising your abdomen, but chances are you didn't do that. And so you wouldn't average, oh, well, this individual went through 0, 1, 2, and 3, so you wouldn't average those all together. You would just say that individual reached 3 versus another individual that only reached 1.
Got it.
Does that make sense?
Yeah, that makes a lot more sense.
Logan, is your hand up for that reason or some other reason?
Oh no, that was way back when. I guess I forgot to put it down.
Okay.
That was on the previous figure. Sorry.
Okay, cool.
Okay, so the previous slide was the biggest thing they did, but does anybody want to walk us through what latency is and what this means according to the results?
Latency?
Yeah, latency.
Just like how long it took them to respond to the stimulus.
And an important thing to note here is it's kind of the weakest measure of everything. It's just seeing the screen and then moving. It's not scoring. It's just their reaction to the stimulus. And what is notable about all of these categories when you compare the two species to each other?
There's no difference between them.
Yeah, that's correct.
And they're finally giving you what?
Standard error bars.
Finally giving you standard error bars. Which, those are standard errors, they're not confidence intervals. You would have to lengthen those standard error bars by about a factor of 2 to get what the 95% confidence interval is. And as soon as you do that, then nothing there is significant.
So this is their phylogenetic results. Table 2 just kind of shows the morphological characteristics that they used when they were constructing their trees and their cladograms. One thing, if you notice down here, it has an asterisk and it says this character was omitted from the analysis and then later mapped. That was the character for the presence or absence of the brushes, tufts, or bristles, whatever you want to call them, on the spiders. So they used 15 of these characteristics to create or to do the analysis, and then they mapped the occurrence of tufts onto the trees. So if you look over here at figure 4, these black bars right here show the presence of tufts, and the white bars show the absence of tufts. So can somebody explain to me the idea and the concept behind A for figure 4A and what the tufts means in this one?
So they're trying to come up with a theory as to why 2 of these Schizocosa members have tufts and then the Floridana doesn't, that member. And so in A, what they're saying is that the trait of tufts evolved in 2 separate instances, so there's not a single common ancestor that evolved tufts, and then every descendant from that ancestor also had tufts, it's showing that they evolved in two independent instances.
Yes. Could someone different explain figure 4b?
This is the reversal where tufts was present in the common ancestor, and then there was the character reversal in Floridana. So they lost the presence of tufts through another evolutionary event.
Yep. So knowing these two things, knowing the difference between the two trees, think about the pre-existing bias hypothesis. Can someone maybe take a stab at which one of these trees they think has the strongest argument or strongest support for that pre-existing bias hypothesis?
I think B shows like the pre-existing bias because from my understanding, don't you kind of need an ancestral like presence to confirm that was a pre-existing bias hypothesis?
So we actually decided that it was A that shows the strongest support for the pre-existing bias because of the fact that it didn't evolve from a common ancestor. And Dr. Klinsky might be able to help me out on this because he was the one that kind of explained it to me. But like Logan said, it shows here that tufts would have evolved for the Ocreata and then again for the Crassipes. Whereas down here, it would have evolved at a common ancestor in which Ravnari kind of broke away.
Yeah, and so put your arrow down at the base of that, Glade.
Here.
Yeah. So right there, all this is really telling you is these are two equally parsimonious, equally valid ways of thinking about trait evolution. Now, if tuftedness evolved in that common ancestor and then was lost in species A in Stridulans and Ravnari, so there's where the common ancestor is, and it's lost in those other three species and retained in Craspes and Ocreata, then that's not pre-existing bias. Now move your arrow about halfway between that common ancestor and the common ancestor to the Ocreata clade. So if tufts evolved at about that point in time, then any preference that Ravnari has is a pre-existing bias. Now go to where Ravnari and Craspes and Floridana all come off of a common ancestor. If it occurred at that point, then there's really not, then that really supports the pre-existing bias because the common ancestor of only Ocreata, Craspes, and Floridana had tufts, and it happened evolutionarily a long time after Ravnari and Stridulans and species A branched off. So because these two hypotheses are equally valid, and you don't have any idea knowing where on that branch from where Ravnari comes off to where Ocreata and Craspes and Floridana all have a common ancestor, because you don't have any idea where on that branch tufts actually evolved, it's kind of hard to make a decision about whether this is an example of pre-existing bias or not. If it happened the way it's illustrated in A, with two independent evolutionary events leading to tuftedness in Ocreata and Craspes, then that's strong evidence for a pre-existing bias in Ravnari. Does that make sense to everyone? I guess it does then. Okay.
So that and there we go.
Yeah, so the biggest takeaway from these phylogenies are that, tufts are an evolved trait. And we just kind of explained it's a response to the pre-existing bias hypothesis. I don't know if we need to talk about that any further. We kind of just addressed that.
I want you to pay attention in the first paragraph of the discussion. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. I guess the 12th line down, it says, the reduced, although overlapping in statistical significance, levels of response seen in female Schizocosa ocreata shown altered video images of conspecific and heterospecific males suggest that the presence or absence of either tufts or species-typical leg waving displays affect mate recognition. So in their discussion, they are basically saying, we think these differences that we see in the height of the bars are real, even though we don't have statistical support for that statement. And so they do it in a parenthetical statement, but they really are hedging their conclusions quite a lot because they didn't have a ton of statistical power to address their hypotheses. You catch me where I'm at?
Mm-hmm.
A lot of speculation.
Yeah, it's way more speculative than I would like to see. I don't want any of you ever to have to write in a paper, the reduced, although not statistically significant reduction, blah, blah, blah. Like you should avoid having to ever do that.
So because of their results shown in figures 1 and 2, those graphs with the spider sitting on top of the bars, they did conclude that the Ravnari females tend to prefer the males with tufts. And this suggests that female preference in Ravnari is a preference for a novelty characteristic. And they discussed that these tufts for both Ocreata especially, but it applies to both species, the tufts might be signal amplifiers. They didn't really have conclusive evidence that it was the tuft that was the female preference, or if it was like the leg waving technique in Ocreata. So they did suggest that tufts could be a signal amplifier for the signal, which could possibly be the leg waving technique.
Yeah, kind of going off what Haley was just saying, the Ocreata females preferred males with the leg waving technique. And what does this suggest? Kind of going back earlier into the presentation about where these species are located, it's suggesting that environment plays a role. Does anybody recall why the environment plays a role in the techniques they use to attract females?
It has something to do with the fact that I think one of them uses vibration and the other is purely visual, or uses both or something like that. I forgot which one.
Yeah, they both use the vibration, but the leg waving technique is more effective for the Ocreata males because of the leaves, the dry leaves in the deciduous forest. It doesn't make sense to vibrate as often as display their leg waving technique, because the females are going to see better than they can hear in that environment. So as I was kind of confused, where you're thinking that one of them doesn't vibrate, one of them does vibrate. Both of them vibrate. It's just the leg waving technique is used more frequently by the Ocreata because it's more useful for them to attract females. Jack, you had something that you're going to say, or you kind of?
I can't remember what I was going to say, but it's all good now.
Is anybody else?
If you're down in really complicated leaf litter, it's hard to be seen. And if you're tapping on leaves, that vibratory signal dies at the end of that leaf, whatever leaf that it is that you happen to be on. So if you happen to be a male spider and you're on the same leaf as a female spider, well, for one thing, you're way too close to her to begin with, because if you haven't signaled that you're the right species, she's going to kill you and eat you. And if you're signaling to a female that is on another leaf, she can't necessarily sense your vibratory cues, but what she might be able to do is she might be able to see your leg waving. And so in the more complicated, complex, three-dimensional environment of a deciduous forest, where all the leaves haven't been flattened by sediments from being floodplained, you have better luck being seen than being heard because of the structure of the leaf litter is kind of their point.
Didn't they also mention that the Robneri probably don't wave as much because it's rather conspicuous and could increase your risk of predation?
Yeah, anytime you make yourself more obvious to a member of the opposite sex, you are also making yourself more obvious to a potential predator. And if you've ever walked around a floodplain forest, what generally happens is when it floods, it puts a layer of sediment down on the leaf litter. It tends to flatten the leaf litter more, and it makes it all kind of a uniform kind of color. And as a result, if you're at the level of a spider that's this tall, your sight lines are much better than if you're in an upland forest where the leaf litter is deep and you might not be able to see something 2 inches away from you. And so you need to be more visually obvious in that complex leaf litter environment than you need to be in a flattened leaf litter environment. Does that make sense, Logan?
Yeah.
Was that Logan or Jack? That was Logan, yeah.
So we're at the point we're going to talk about the future experiments, and we wanted to hear what your guys' ideas were for furthering this research.
I talked about a more complete phylogeny where you could like better understand where Robneri branched off and where tufts evolved, and then better kind of answer this pre-existing bias hypothesis.
I also said that when we discussed with Dr. Polinsky, he pointed out that the phylogeny that they created was largely influenced by like a different researcher or scientist, and they only used those 6 species when there were way more to include. And I think you guys mentioned like the updated paper from 2005, but he showed us that in that phylogeny and how much more updated it is, and that would tell you a lot more about whether pre-existing bias was a thing or not. So that was also my future experiment, was to basically create an updated phylogeny where all the species of Schizocosa are included in the exact point at which the tuft trait evolved is known. And then you can make those conclusions about pre-existing bias.
Yep. So, Luke.
Yeah. So we all kind of dove into the phylogeny. They need to be updated, to be more constructed. It's not super revealed here. A lot of speculation has to be made off of those things in this paper. But another thing too that they mentioned was this whole confirmed the leg waving is the female S. acreata preference. And I don't know if you guys recall, but they said that female choice was something that they didn't really feel like they tested clearly in the results. Does anybody recall why? It's kind of a specific exact question, but does anybody recall why? Like the role of tufts with female choice and what they can do to get a clearer result as to how that plays a role in female choice. What you can do to tufts, rather than enhancing them, what's another thing you can do to tufts?
I don't think anyone else keyed in on this, Luke.
Yeah, it was.
They mentioned like taking the tufts away,
Yes.
like making sure that like the heterospecific species had tufts, but like S. acreata females with S. acreata males had like no tufts, or just like the controls.
Yeah, so reducing tuft size and enhancing tuft size, and showing females these video images of these males with these either reduced or enhanced tufts will kind of give you a better idea of how these tufts play a role in female choice. Something they did not do in this study. Kind of another avenue to take some of the things that weren't as clear in this study into the future, aside from the phylogeny.
So then building on the phylogeny idea, we talked about doing, instead of morphological phylogenetic testing, using molecular data in order to do the phylogenies, especially since at this point in time, there's been a huge advancement in technology and we have access to doing molecular testing. And then Dr. Kolinsky discussed with us the possibility of using a different species. So they used Ravneri and Ochreata, but if you look at this phylogeny, which is from Stratton 2005, both, here's Ochreata right here, and then Ravneri is up here. And if you look, they still have a similar, same common ancestor. So we discussed using a completely different species to look at the evolution of tufts and female preference. And his suggestion was to use S. saltatrix right here, because it's a better choice since it lies outside the Ochreata clade, unlike the Ravneri species.
Also, and you could use any of those basal groups that don't have tufts. There's another thing that I want you to pay attention to in this particular phylogeny that is different from the McClintock and Utz phylogeny. What else do you notice about this phylogeny?
That Ravneri lost tufts because of a reversal, and tufts were part of their common ancestry.
Well, okay, and that's what Haley just referred to. Haley, go back to the phylogeny from the McClintock and Utz paper. Who's in the untufted branch over there?
Up here or over here?
The untufted branch. You have species A, whoever that is, Stridulans and Ravneri.
Mm-hmm.
Go back to Stratton's paper.
They say that Stridulans is
right
tufted.
here. Yeah, they have.
Stridulans is tufted.
Am I wrong?
And it even has a little picture of Stridulans tuft. Now, it's not as tufted as Ochreata and Crassipes is, but it's got a black tibia and it's got kind of intermediate sized tufts.
Why is it that we read the other paper and not this paper?
Because
like the horribly unspecific, we don't know anything paper, and this is a beauty.
okay, so one of the things that Stratton doesn't do is Stratton doesn't do any behavioral testing. And the reason that I have you read the McClintock and Utz paper is that it illustrates why making conclusions on incomplete information is not really a good thing to do in science. And you go to press with the information that you have, not the information that you wish to have. It's kind of like Donald Rumsfeld going to war, for those of you who remember that. You go to war with the military that you have, not the military that you wish you had. Well, you also go to publication with the data you have, not with the data that you wish you have. And so this just illustrates why, it illustrates a couple of points. It illustrates why phylogenetic systematics is something that is useful and beneficial. And it also illustrates why when you create a phylogeny, you should make that the most complete phylogeny that you can, because doing a phylogeny with a reduced suite of species can lead you to the wrong conclusions. If they had had this phylogeny, their conclusion would have been that female preference for tufts in Ravneri is not due to a pre-existing bias. And also the way in which they equivocate in the discussion and don't ever really come down firmly on saying, you know, this is what we think is going on, is partly due to the fact that they knew that they didn't have a complete phylogeny at the time. What's the challenge of building a phylogeny in this group? It's open to anyone. Anyone? Bueller?
Can you repeat the question one more time?
Well, what's one of the challenges of doing a phylogeny with this group of organisms? You can find it in the method section under the section called the species.
I don't know if this is the answer you're looking for because I'm still pulling up the paper, but I know you mentioned something about Crypsis, and other spider species look very similar to wolf spiders, so that's why the molecular testing would be important.
On page 168, Schizocosa ocreata and Schizocosa ravneri are wolf spiders common to the deciduous forests of the eastern United States. The two species are geographically sympatric, but generally occupy different microhabitats. They are virtually identical with respect to body size, genital characters, and color. And so this is a group of organisms that is evidently recently evolved, recently radiated, and as a result, there are what we call cryptic species, species that are morphologically identical, but are separated from one another either by reproductive incompatibility or by behavior. These kind of sexual species recognition signaling mechanisms that tell the member of the opposite sex that you're a member of their species rather than being a member of somebody else's species. And so that's really commonplace in this particular group of spiders. And as a result, building a phylogeny that is based on morphology, that phylogeny is also going to have problems because you have species like ravneri and ocreata whose females are virtually identical and the males are virtually identical also, the only difference being the structure of the first leg. And so that makes things difficult, which is why in your suggested future research, doing a molecular phylogeny might be a better way of going about getting at phylogeny in this group.
Yeah.
So McClintock and Ewitz didn't do a phylogeny because, for one thing, they're behavioral ecologists. That's not really in their wheelhouse. And they would be trying to attack a phylogeny that is already going to be challenging to construct if all you're going to rely upon is morphological data. So they didn't do it because it's hard, and that's a perfectly reasonable reason to not do something. But as a result, they published a paper that has, as we know now, limited utility because of the incomplete information.
Oh yeah, another reason to read this paper is kind of a kiss-up answer, but it really embodies the Bloom taxonomy, evaluate and create part of the class.
Oh yeah, sure. I mean, I think one of the things that I sometimes do that I shouldn't do, which is just give you a bunch of really good, well done science to read, and then not ever show you how science makes mistakes and how science could be done better. There's as much to learn from really good papers as there is from papers that have flaws. And this is just one paper that has quite a few flaws.
I think that's the last slide.
Yeah, that's the last slide.
Okay. Any other questions for Luke and Haley? And it's time. Alexis asked me to hang around for a minute, so the rest of you are free to go. We're going to be talking about sexual conflict on Wednesday, and then we don't have class on Friday because of Good Friday or Monday because of Easter Monday. And so I'll see you on Wednesday, and then I won't see you again until the following Wednesday.
So do we just turn in all of our stuff to Moodle? Is that, or we just email you? Or
No, just email it to me. I'm printing things and reading them and doing. I'm actually, Jack, you'll be happy. I'm doing more things on virtual versions of things now than I was before.
Good. There you go.
So,
I'm going to ask this. Did you want them as PDF or as a Word document? Do you care?
I guess I don't really care. I think Word documents are a little easier for me to make digital annotations on, but I can make digital annotations on either.
Okay.
Okay. Thank you.
All right. Oh yeah. Last thing. What up with all the passive voice? Get rid of it.
Are you talking just in methods, or is it like all throughout stuff?
It cropped up more in methods, but for some papers it was throughout. But yeah, most of you probably had it in methods. And passive voice is bad because it takes the object of the action and puts it as the subject, and so it makes unclear who's actually doing what. And so
Gotcha.
when you talk about, well, with coral snakes, when you talk about the models were attacked, the implication is that the models were attacked by avian predators, but the models were also attacked by non-avian predators, and occasionally they just got stepped on by hikers. So it's
I think my problem, I mean, I didn't really have a lot of passive voice in the paper you just sent me, but I just notice it like when I'm trying to write in general, specifically for literature summaries, because we're condensed to 2 pages, is that I have a lot of information that I want to put in 2 pages, and I can't write like McClintock in whatever 1996 did this, and then they did this, but we also can't use they. So then I have a problem.
Oh no, you can use they.
Words.
You can use they anytime that it's clear that you're continuing the discussion of McClintock and Ewitz.
Okay.
And how do I know that you're talking about McClintock and Ewitz?
You just referenced them.
Summary about the whole thing.
Because that's what the whole summary is about. So introduce them at the beginning, and then you can use they over and over again, as long as it's clear that you're talking about the researchers, rather than, for example, talking about the spiders they.
Okay.
So you can do that just fine. Even within a paragraph in like a literature review, if you're devoting a paragraph to a particular study, you only have to mention the authors of that study at the beginning, and you can continue to refer to them with a pronoun, as long as it's clear to me when you're shifting out of that study into another study.
Okay.
Yeah, I've never dinged anybody on using pronouns as long as the reference to the pronoun was obvious.
Okay. Cool.
Yeah.
For me,
And the other nice thing about active voice, Kennedy, is that it actually takes fewer words to write in active voice. And so if you get in the habit of writing in active voice, and you didn't have much problem with passive voice at all in your paper, you can actually say more things in fewer words with active voice. So you can squeeze more of that detail that you want to squeeze in there into there.
Mm-hmm.
Somebody else was about to say something.
Yeah, I had several instances of passive voice in my paper.
Oh my God, you did.
I was right on the brink. And the reason I did that is because just based on English writing, English classes that have taught me how to write, it just gets repetitive if you use the same pronoun over and over and over. So I'm saying they did this, the researchers did this, they did this, the researchers did this. I feel like that gets repetitive and redundant. So that's why I feel like I trapped myself several times going with passive voice. So I just don't want to be repetitive, because that's a faux pas in writing, is being repetitive and redundant.
So 2 things. One, you're not in English class, you're in science class.
Good science writing requires good English writing, though, which is why grammatical mistakes are part of this contract grading.
Yes, I know. So the second thing is, instead of writing those in a series of short sentences, they did this, then they did this, then they did this other thing.
Mm-hmm.
Say they did this and this and this and this.
Okay.
It also can't have run-on sentences.
Well, that's not a run-on sentence. It's a complete sentence with a compound verb. You've got multiple verb clauses strung together.
You could also try a gerund as your subject, like a phrase with an ing verb at the end as your subject. That's helpful for me.
Whip out the gerund if you want. Haley's smirking about the gerund. Yeah, so there are ways of writing in active voice that is not redundant, but I see your point, Logan. So you just need to one of the complaints that people have about my writing, and they also have it about my speaking, is that I tend to write in really long sentences. And part of that is, over the years, I've strung verb clauses together because this person did this and that and that and that and the other thing. And while you may not want to talk that way because you lose your audience because they stop paying attention to you because it's just too long of a sentence now, you don't necessarily lose people when you're reading that because you can always go back a little bit and say, okay, what did they do? They did this and this and this and the other thing. So,
Okay, got it.
it's especially problematic in a literature summary because you're referring to the same people all the time. You don't get to break it up with different names. But a literature summary is a very contrived sort of writing task. And so I would expect you to be able to avoid passive voice better and less redundantly in something like a literature review because you're going to be talking about lots of people's work. Does that make sense?
Yeah, that makes sense.
Okay, cool. The rest of you, yeah, Luke?
I don't think you sent me my literature summary. I don't think I got it. I looked in the email.
Oh,
I know we had like a little conversation on writing, but I don't think it was in there.
okay. I'll see why it didn't send.
Okay,
Honestly,
just,
I immediately deleted it this morning because I didn't know. I just thought
It
it was
looked like
spam, so I have
spam,
to go
yeah.
back to my trash to get it.
Okay.
But
All right. Yeah, I'll resend yours as well. Let me make a
I
note.
have mine. I'm good, but I just thought maybe he made the same mistake that I did. But
Okay. All right. So you still have yours. All right.
Yeah.
I'll figure out why Luke's didn't send, and so
I don't know why either.
I'll drop you an email when I. Sometimes things get caught in the ether. Like I've done scans for my own use, and then I come back to my office, and they should, in theory, be in my mailbox, and they appear like 2 hours later, and I don't know what happened to it in the interim. So anyway, I'll look into it, and I'll just resend it.
All right, when you get around to it, that's fine. Thank you.
All right.
thank you.
All right.
Yeah, thanks.
Later.
Alexis, do you want me to stop recording or keep recording?
You can stop recording.
Okay, I'm